You're listening to Body Banter, a podcast where we have conversations about the human body in all its forms and from as many perspectives we can find.
We are your hosts, Shagun Yedile
and Claudia Krebs.
And we are professors of anatomy in the Faculty of Medicine at the University of British Columbia in Vancouver, Canada.
Anatomy is for everybody and every body, and we're here to get the body banter going.
We hope you enjoy this episode. Hello, and welcome to this episode of Body Banter. My name is Shagun Yedile, and I am calling today, speaking to you from Kelowna, which is in the traditional unceded and ancestral territories of the Syilx Okanagan Nations, otherwise known as University of British Columbia in Kelowna. And as usual, I have my co-anchor, Claudia. How are you?
Hi, Shagun. I'm doing well. I'm joining you from Vancouver on the traditional, ancestral, and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh Nations. And we have a guest here. Chris, welcome to the podcast. Why don't you introduce yourself?
Thank you so much, Claudia and Shagun. I really appreciate the invitation to come and speak to you all today. So my name is Chris Smith. I'm a postdoctoral fellow in the natural history here in New York City. And I work in the division of anthropology, but I focus most of my research on sort of the biological side of things. And most of my research focuses on the evolution and evolutionary change in the inner ear in humans and cranial structure. So my position here is primarily research-based. So my day-to-day is sort of driving a lot of my own research initiatives and also collaborating with others here at the museum. And as I mentioned, looking into inner ear evolution, dealing with the organs for balance and hearing, and other parts of cranial structure.
That's so cool. So that sounds like a dream job, right? Working at the Museum for Natural History in New York City, working on your passion piece of the human body, the inner ear, looking at hearing and balance. How did you get there?
Yeah, I mean, I will say I enjoy my job. I love coming into work every day. So I kind of have quite a convoluted journey to get to where I am now. And that really started off as an undergrad at Salisbury University, which is a small college on the Eastern Shore of Maryland. And so while I was there, I majored in exercise science. So I was really interested in just the human body in general. My initial goal was to become either a physical therapist or athletic trainer. But while I was there, I sort of reignited my love for art and illustration and wondered if there was a way to combine the art and science. So then I looked into graduate programs in sort of biomedical communication. And at that time in 2010, 2011, there was sort of 4 accredited grad programs that I found for biomedical illustration. And I thought that was a great, way to sort of directly combine science, both science and art. And I reached out to one of the programs, Johns Hopkins in Baltimore, went to go visit and realized I needed much more art training. I really had no formal art training. So after I graduated undergrad, I went to go take a year of fine art training at the Schuler School of Fine Art. It's like a small atelier or sort of classical setting for teaching art in Baltimore as well. So I spent a year there, just learned traditional methods, grinding my own paint, grinding my own pigment, doing sculpture, all of that. And then finally got in to the program at Hopkins, did my master's there in medical illustration. And that program, you basically start out doing traditional drawing, painting, then moving into digital media, and then other aspects of animation and modeling, et cetera. And so I had a great time during that program, getting a taste of what sort of modern biomedical communicators are all about. And then following that, I started working as a full-time medical illustrator at the Icahn School of Medicine at Mount Sinai in New York. So that's kind of how I wound up from being a Maryland native then hopping into the middle of New York City. And so there I worked under Jill Gregory, who was one of the medical illustrators there as well. And so Jill and I worked as the team for medical illustration. I was there for about 2 years. And while I was there, I also got in touch with Jeff Laitman and Joy Reidenberg, who we're all very familiar with, and spent a lot of time in their anatomy labs doing illustrations and modeling for the anatomical education curriculum, as well as some of Joy's whale dissection work, which was a lot of fun. And while I was there, I realized that I did have a really strong interest in human origins and anatomical research in general. I kind of wanted to try my hand at doing and driving some of my own research, focused on a lot of my own interests. And so I approached Dr. Laitman and asked if he could if I could apply for a PhD program through the City University of New York, where he's adjunct faculty. That's how I got him to do my PhD through CUNY. And I think it was last May, I got my degree and then switched over here to the museum for my postdoc. And sort of all along the way, I always try to find ways to integrate a lot of my artistic training and visualization background into the research itself. And there are quite a few avenues to do that, ways to do that. And I think I found many possibilities for allowing the actual visualization to either open up new questions or drive new avenues to approach anatomical research itself. So sort of finally a long journey of education in and out of school. I'm now here based at the museum, primarily doing research, and we'll see where it goes. That's how I've got to where I am now.
That is a fascinating journey and convoluted, like you said, but it's really fascinating how exercise, sports science changed into art, and then now into function relationships, and how that drives visualization and how that helps communication in science and so on. That's amazing. It's almost like you kind of like an a la carte. You design your own research. You picked a little from here, a little from there, and combine it into the perfect work you're doing right now. So that's really great to hear. I guess my follow-up from there would be, do you think that that background has prepared you for what you do today? Because a lot of people who graduate with science degrees and so on, end up in careers that maybe they're not so happy with, or that's just, they find that it's not really what they wanted to do. But how do you see that background with a rich science base, and then combined with art and visualization, communication in science? How has that kind of formed you into what you're doing today? Could you speak to that?
Yeah, of course. It's a great point that I think my non-traditional background, convoluted journey, has helped me in one of the best ways it's helped me is being flexible and creative in how I approach research questions or science questions. And I think that really is owed a lot to my sort of initially artistic training, and then just science communication in general in my master's program, because there are a lot of topics and questions in either human origins research that I was, that I'm involved with, but also just comparative biology, that on the surface seem like they're already answered, well addressed. There's really no need to go back and do more research in, but coming at it from a perspective of either like questioning the paradigm or thinking critically about like structural relationships in anatomy, or new ways to visualize structure, has opened up new doors that I didn't initially expect. And one example of that is in my research into inner ear structure. We think a lot of, parts of the body are already fully explored and understood. And one of the areas of the ear, sort of at the center or the core of the labyrinth in between the organs of balance and hearing, there's a lot of membranous structure that's important for support structural support, but then also like things like fluid movement and transfer of the fluid inside of the ear to different compartments. And so, like, one initial way that my research and thinking creatively, I think, helped in that area of research specifically is thinking critically about that specific membrane, the membranous structure, and kind of challenging the paradigm of what we think we know about the morphology at the core of the labyrinth. So a long-winded way of saying that I think thinking creatively and being flexible from sort of a artistic or creative background, I think helps immensely in science.
I love that. There's an old adage among anatomists, you only see what you know. You've turned that around in that you know what you see because you have a different way of looking at the structures through your arts background. How did your supervisor, Jeff Laitman, react to that? Because he is, of course, an icon in the field, and also probably, I don't know, like not used to possibly this approach, right? Of seeing to gain knowledge, whereas we're trained to gain knowledge and then find the structures and see them. So you're almost going back to like ancient anatomists, right? Who were really in that discovery mode. I want to get more inside of your head of how you see and how you think. Please do share.
Yeah, of course. That's a great question. And interestingly, Dr. Laitman was one of the driving factors in pushing me to really think outside the box in terms of using visualization to drive the research. Because of the fact that, especially in the ear and the areas of the ear, and I think that's why I gravitated toward that area for research, because it's so, it's a complex structure and difficult to visualize. But he was always very supportive of sort of just getting in there in terms of, so I worked a lot with like micro CT data, where there were, we had specimens of temporal bones that were stained and then scanned to visualize soft tissue. So kind of just getting in there in the world of the ear, as he would say, and looking around, exploring, and without really any preconceived notion of exactly what I was looking for, like having an idea of how my dissertation was taking shape. But using various software packages for both like reconstruction, segmentation, and then having like a large enough screen where you can kind of just zoom the camera in and sit inside of the structure you're looking at. And by doing that, that sort of gave like a step-by-step roadmap into trying to understand the relationship between hard and soft tissue in this part of the ear, which sort of formed the basis of my dissertation. So, I would come up with these reconstructions of the anatomy and what I thought was going on. And it was like every meeting I would meet with him and he'd be like, yeah, that sounds great, but, really think about this or kind of go back and question that. Like why, and even think about the terminology, when we think about the ear and the vestibular system, it really derives from the word vestibule, which really just means entryway. And for, millennia, it was thought that the ear was primarily for hearing, and the area that I'm interested in and also where the canals are was sort of accessory to hearing. And that's why the vestibular system, which is really for balance and 3D spatial awareness, is really just named after an entryway. And so thinking critically about that visualization, but then also how terminology shapes our preconceived knowledge was something he really championed and helped formulate how I approach even research today. It's been very eye-opening.
That's why he's one of the best, right? Because
I
he
agree.
does
Yes.
open those doors and encourage people to go find their own way. It's not a coincidence that you are applying your 3D understanding and 3D visualization to the organ that helps us live in a three-dimensional space, right?
Yeah, definitely,
So,
of course.
tell us how your own understanding of how we perceive three-dimensionality and how we live three-dimensionally, and your ability to visualize and see beyond what is known, how that all worked together.
Yeah, I think it's interesting because the system that I'm interested in studying is essential for how we move and detect and are sensitive to gravity and our 3D spatial awareness. It's interesting thinking about the function of these organs, because as you know, you're going through and I'm approaching things straightforwardly and reconstructing the actual anatomy. The morphology and the anatomy is important for their function. And so it also adds another layer of sort of research inquiry into asking yourself what differences in morphology of different parts of anatomy may relate to function, which relates to 3D spatial awareness. It brings up another question of how the organs are configured within the head, because that's also an important structure or an important aspect of their function. And so, and then even beyond that, how these this organ system or these organ systems function during movement. So during movement of the entire body, it's almost I've have found myself both confused and excited about new ways to try and understand how the anatomy, the functional anatomy, behaves both within the rest of our body, within the head, and during specific movements. One of the challenges in approaching this area of anatomy is that a lot of studies are, in essence, in isolation on the ear, because we have to really zoom in at high resolution in isolation. But it's part of the cranial structure, and it's part of how we detect, how we are sensitive to 3D spatial awareness. And so you really have to have sort of a holistic and comprehensive understanding of the ear in context at multiple levels. So it's extremely challenging, but I think with new imaging methods and new technologies, we're starting to get at how the ear and different levels of its anatomy are configured within the head and how they're functioning at many different levels. But it's always sort of a conceptual challenge to try to pull yourself back so you're not, I spend a lot of time inside of the ear in minuscule structures, but they're extremely important in movement and posture and 3D spatial awareness. It's just sort of a multi-leveled problem, but fascinating. So I'm always learning more every day.
Absolutely, I love that connection, and the conceptually, it's really mind-boggling how you're working in a small, tiny area with three-dimensional structures that enable us to live in this wide open world in three-dimensional spaces. It's really fascinating. At the risk of getting a little technical, I'm curious about how technology has kind of improved your ability to see and to visualize the structures that you work with versus normal like pencil or pen line drawing and just normal just pen and paper. Is there like a weight cost benefit thing there where some tools work better than others, and how you can use simple tools to answer complex questions versus using more complex tools to answer profound questions? So how does that work?
Yeah, that's a great question, and I guess I can answer that from as a two-parter. On the one hand, related to visualizing the anatomy, I think obviously computer imaging, digital imaging, medical, CT scans, MRI, all of that has enabled our study of the inner ear to basically just be possible recently, because prior to that, it was all microdissection work and observation. But in the 20th century, and more recently with the advent of like micro CT, synchrotron, it's really opened up a whole new world into visualization. And then building off of that, I utilize a lot of like segmentation from the imaging to create digital models and meshes to capture the actual structure with really no artistic hand involved. And then oftentimes in my research, what I'll do is I'll use that as a basis to do hand-drawn either illustration, digital painting, or modifications to highlight certain structures or processes. But I try to keep as much of the real data as possible. But yeah, very often I'll do additional work, and I try to keep it as traditional feeling as possible, because I always like to have the hard to explain, but like that traditional feel in the artwork. And oftentimes I will opt for more clarity and simplicity in delineating structure. Sometimes I like to go more into detail of the anatomy, but also depends on the purpose and the audience of the piece or the paper. On the other hand, in terms of the actual like statistical analyses that I do for my work and shape analyses, that has a similar, I think, workflow with both using more complicated methods and simplified methods. And a lot of that has to do with, like, for example, one of the questions that I often come back to is, has there been shape change throughout an evolutionary time period, or are there morphological or shape differences among populations? One of the challenging things in the area of the ear that I work in is that the bony structure is very like amorphously shaped, if that's a word. There's not a lot of bony features that are identifiable. And so to enable as much interpretation of like the results as possible, I oftentimes always go back to a simple landmark-based approach, rather than there's a lot of sort of landmark-free methods available now, analyzing topology and doing heat mapping to look at slight differences in structure. But the area that we're looking at has so many aspects of shape that can be caught in those analyses. I always try to be as simplified as possible, so that the results we get are as interpretable as possible. So I think whenever I do statistical analyses on shape in this part of the ear, I always opt for more simplicity, especially at this stage where it's more of an exploratory nature. There's always a time and place for complexity versus simplicity.
Yeah, I that's really a great point that you introduced, that you brought there, that it depends on the audience and how you would like to, keep things simple, depending on the purpose. When it's statistical analysis, then you can get into more complex, things and complex imaging but rather if it's like for a simple audience, you would keep it simple. We see that as well in our biomedical visualization course that Claudia and I teach in, how students are really they need to know the audience. You need to know with the audience that you're speaking to be effectively communicate with them, and that's really important for many areas, but particularly though for science communication. Maybe shifting to a different topic to maybe to ask, maybe personally, have you seen yourself changed? How have you seen yourself evolve? Because you've been on this journey that is really fascinating. And have you become a keener observer? Have you become someone who's a bit more technical or more analytical, or maybe a combination of all of this? What do you think?
Yeah, that's a great question. I agree 100% that I have changed in many aspects. And I think it's a testament to the different types of training I've had at different intervals. So going from very classical fine art training to, the hardcore statistical methods in my PhD program has given me different insights into how, like, for example, to think more creatively or more flexibly, like in the illustration program, by learning multiple different programs quickly, adapting to whatever the project needs, and then becoming, especially in my doctoral program, more analytical and being very meticulous in like data management and collection. And even that has applications into my sort of like my hobby time for my actual fine art in terms of organization and planning. When I was in fine art school, I didn't pay much attention to sort of planning out the composition of pieces because I was just more focused on learning the techniques. But now, having more like analytical training in general, I maybe unconsciously have become much more of a planner, and so that's leaked into my art life as well. But I think everything that art and science really have a lot of crossover, and in terms of how we approach problem solving, and then how we disseminate knowledge. And it's been a very interesting journey, but I think it's definitely given me areas and filled in the gaps to approach things, like I said, sort of a more creative and flexible way that I didn't think possible initially.
That's so cool. I love this combination of sort of the analytical discipline and then the creative flexibility, and how the two come together to really create knowledge that wouldn't have been possible without the combination of the two. So congratulations on that.
Thanks.
So,
it's very unexpected, but it seems like it works.
I love that. And I wonder if that's really a paradigm that we should be teaching all science PhD students, right? I feel sometimes folks get boxed in with their methodology and with sort of a classical approach of academic discipline and disciplinarity. And maybe we're losing some of the plot there, right? By ignoring some of these different ways of knowing and cross-disciplinary approaches.
Yeah, that's a great point. Yeah, I think that I think in many instances, students who are going through scientific training related to research are, in some aspects, under time constraints to learn the techniques to figure out a dissertation, learn about project management, and come up with a feasible way forward. But I do think it is valuable in either reiterating the creative flexibility or ways to approach problems that allow those to think outside the box. It's just a matter of if that would be effective as an additional course, or if that could be something that's just worked into how we teach how, like, the scientific method works, for example, or how we teach students to approach it. It's difficult to sort of package and teach the students, because I honestly feel like a lot of the experience that I have too has come from having to problem solve, when it's like late at night and I have something that I need to get done to send out to other co-authors, and I have to think quickly and on my feet to be able to pivot. And I mean, I think that that's a natural skill that many scientists learn overall. But I think that taking some of the teachings of sort of like biomedical communication or art could be helpful in achieving that flexibility sooner. It's definitely a great discussion to have, I think, particularly in, for example, your courses and others who are teaching biomedical communication. But then that could even form a bridge to have more sort of hardcore science departments take note as well.
I totally agree. Okay, so we've talked a lot about what's going on in your head and your methodology and how you've combined these disciplines. I want to get back into the inner ear. What is the finding that you have? What did you discover?
Yeah, so great question. So I would say the main thing that came from my dissertation work was that this area of the ear that's very hard to visualize and conceptualize, the area that is sort of the bony house of the otolithic organs, which are our gravity sensors. It was always thought that there wasn't really a relationship between that outer bony house and the internal organs themselves. Our research has shown that there is a relationship, and that relationship can be captured via certain bony features on the bone, which now enables us to study that area of the ear across at least mammals, because these features are shared across mammals, back in time because they're also apparent in fossils as well. And the sort of next step that we're currently working on is to dive a bit deeper so that now that we can analyze and assess morphology of this region of the ear, is this area of the ear really a part of a single peripheral vestibular system along with the canals? And it's sort of just one conglomerate of organs that deal with our sense of balance and 3D spatial awareness? Or is it more modular and that these organs inside of this vestibule sort of bony, blobby house area, is it possible that they are sort of their own thing, their own entity that has retained evolutionary distinction even up until humans? So our current work is sort of investigating that. We're still working on the bony anatomy, but now that we're getting more and more data on other animals and the soft tissue, because there is some interesting evidence that shows that this area of the ear isn't just sort of accessory to the canals or just part of one single balance system. There's more complexity going on, at least anatomically. So there's much more work to be done, but very, very exciting for the path forward.
That's science, isn't it? We always say there's always much more work to be done.
Exactly.
That sounds really fascinating. How you've discovered that the otolithic organs themselves are actually, there's like a functional relationship with the containing bony cavity in which they are situated. That's amazing. And I'm wondering, not to give you more work, but a future direction would be, how about in zero gravity environments? How about, where astronauts and people like that who are trained, and have spent months and a long time in zero gravity environments, is there like a difference? Is there like an evolution or change in their middle or inner ears? Have you
zero gravity zero gravity is a very interesting sort of environment to assess inner ear function. And for many years, NASA funded quite a lot of projects on inner ear structure and function. In terms of the anatomy and the morphology, I wouldn't expect there to be really much change, but there's definitely differences and changes that go on neurologically, and how we adjust for like the absence of any sort of stimulation on things like the otolithic organs. But understanding how other aspects of the anatomy, the hair cells, the arrangements, the otoconia, the calcium carbonate crystals that overlay those organs, is there differences in how they're being displaced in zero gravity? There is still more work that could be done on that area. The difficulty is trying to visualize those types of structures in a living person who is in zero gravity, without having to do that post-mortem. So yeah, it's always the difficulty, because this area of the ear, and the knowledge we have from it in terms of its anatomy and structure is really only from cadaveric material or post-mortem. And so there could also be other aspects of the ear that in living populations that we aren't aware of, because we also haven't gotten to that level of visualization yet. But yeah, the zero gravity thing is definitely fascinating.
That's amazing. And I completely get it. It's true that it would be difficult to monitor changes in someone in a zero gravity environment while they're going about doing other things, and you have to rely. But you can tell. I mean, you've already started working in this area. There might be others who get an idea about maybe how to do that. And so we gradually get into that stage in our discussion where we begin to ask you about your own favorite anatomy, in terms of your favorite body part. This is body banter after all. And we'd like to ask our guests, have you, in all your work and all your experience, all your research, or even just personally, do you have a favorite body part?
I do, and I think I'm somewhat biased on the ear, but I will say, other than the ear, in general, the brain is just an extremely fascinating structure. Sort of just the brain and neurological tissue is probably my favorite organ system of the body. It's great. I always remember the first time I took anatomy, and we did the brain removal, and it was just sort of the most emotional aspect of taking human growth anatomy, just because of the impact of seeing in physical form, someone's essence. So yeah, I would say ear and brain.
And they're closely related to each other. I agree that
Of
lab
course.
where
Yeah.
you retrieve the brain is transformative for our students. I don't know, Shagan, can you think of a lab that has an equal impact? Maybe the first anatomy lab.
Yeah,
Okay.
it's always a very, like you say, an emotional, almost spiritual experience for our students when they are able to actually remove the brain and hold it in their hands for the first time. So yeah, great choice, Chris. That's good.
Of course.
Yeah, you won't get opposition to loving the brain in this room. So, so what body part are you not so fond of? Like where you're like, meh,
Yeah, it's
favorite.
let's see. So I would have to say probably the gut in general, intestines, small intestines. And the only reason I say that, I mean, it's a fascinating system, and I have nothing against it. But during anatomy, and also when I was working with Joy Weidenberg a bit on whale stuff, anytime we would work on gut contents, it was just probably the worst smell and most uncomfortable I've been in the anatomy lab. But that's really the only thing I can think of, because really no other structures have a strong aversion towards. It's just yeah, and it's also a system that's difficult to visualize because it's lots of soft tissue. But if I had to pick one, I'd say maybe the gut or intestines in general.
Yep, I can see where you're coming from there. It's interesting that you say it's hard to visualize, right? Because it's a lot of soft tissue and it's around all the time. And I think, now that you've said it, it's like, yes, obviously. And when I think about teaching the gut, it's true. Like one of the key challenges that we encounter is having students understand how things are arranged in the peritoneal cavity. The sort of three-dimensionality of it, how some structures stay put, others move around, and how development kind of twisted everything into its place. That's hard. So yes, yeah. I hear you. Yeah. So thank you so much, Chris, for sharing your insights there. I think your key approach of combining like that flexibility and open-mindedness of the creative world with the analytical discipline of what we call hard science, has really yielded fabulous results, and I'm sure it will continue to do so as you look at this structure in different mammals in your current position at the Museum of Natural History. I am looking forward to a future where more scientists have your background so that we can really build this knowledge in a new way and build in new perspectives.
Well, thank you so much for Claudia and Shagun for having me on today. And this is my pleasure. I'm always happy to chat about ear and art and science, and I think it's been it's been great getting your input also on this kind of sort of interdisciplinary work. So thank you so much again.
Yeah, I want to echo Claudia's thanks, Chris. Thank you so much for your contribution to science, your contribution to visualization, to art, and to our understanding of the evolution of the inner ear. If our listeners would like to read up more about your work, where could they find it?
Yeah, so I have a profile on what's called ResearchGate. It's sort of a social media, not necessarily social media, but a website geared towards showcasing research. So on ResearchGate, if you search for just Christopher Smith, I should pop up. I also have my professional illustration website, csmithstudios.com. And connected to that, which will via a link, will be my new research website, which will be live hopefully sometime this year. But yeah, always happy to chat about art and science.
Thank you so much. And we will link to all of those websites in the podcast description so that people can just find it through a click.
Great.
Thanks so much, Chris, for everything. And that wraps up another episode of Body Banter. Thanks so much, everyone.
Bye, everyone.
Thank you.
Thank you for listening to another episode of Body Banter.
We are Claudia
and Shagun.
And we look forward to having you join us for more conversations about the human body next time.